In the final part of our lecture, we're going to look at the primary lines of evidence that support Darwin's theory of evolution. I'm going to try to keep this video short because I realize that you have 6 videos in this series. When I teach this section and when I test this section, I tend to focus more on the process of evolution and Hardy-Weinberg adaptation, things like that, and focus less on the lines of evidence for evolution. There are some really important things in here that we will talk about, and I will definitely be testing on this, but the bulk of the focus of this chapter is going to be the beginning 3/4 of the chapter and a little bit less focus here on these few ideas. So one of the lines of evidence that supports evolution, and one of the things that Charles Darwin used in developing and fleshing out his theory of evolution, is the fossil record. So the fossil record demonstrated that organisms were different in the past. It demonstrated that there were organisms that lived in the past that no longer survive, so it showed evidence of extinction. And it also showed that as organisms progress and we see fossils that are closer to living organisms, they become more similar. So those fossils that are younger fossils are going to be more similar to living organisms now, and those that are older fossils are going to be different. Now, the fossil evidence suggests that organisms can change over time. Or at least it suggests that there have been different organisms throughout the history of life on our planet. So fossils are basically the remains of living organisms that have turned into stone. So through a process of weathering and sort of natural preservation, hard structures in living individuals have been turned into rock. And so those features are basically images in stone now. And so if we look at the fossil record, you can use it to see the emergence of different traits throughout the history of life. You can see the emergence of our big categories, our bacteria, archaea, and eukaryotes. So even bacteria can leave fossils. The structures that tend to be fossilized the best are hard structures, so things like bones and teeth, claws, whereas soft structures are less likely to fossilize because they basically decay before they can go away. So things like fur, feathers, muscle tissue, things like that, don't usually fossilize. Now, there are some instances, this example here of the Archaeopteryx, where the feathers did actually fossilize. So that's kind of a rare event. There are some problems. Actually, I'm going to hold off on that. We can use fossil evidence to reconstruct lineages and to follow the development and progression of certain traits within particular individuals. And this allows you to see the relationship between organisms, even though you can't see a living common ancestor. So one of the things that's cool about the fossil record is you can use it for to be able to put a date on the organism. Now you can do relative dating. You can say, okay, this fossil appears higher in the rock or in the sediment than an older one or than a different one, and therefore it's younger and the deeper one is older. And that has to do with the way that sediments are laid down on top of organisms. But that's all relative dating. You can only do that, kind of before or after. But you can also do something called radiometric dating. Now, radiometric dating uses something called radioactive isotopes, which are chemical compounds that will over time sort of break down and turn into other things. And they do this at a regular rate, and different chemical compounds decay at different rates. So the most common one is radiometric dating using carbon. Now, there are several different forms of carbon. There's carbon 12, carbon 13, and carbon 14, and this has to do with the molecular makeup of the carbon atoms. And basically, carbon will sort of decay into another version over a set period of time. Now, there are other molecules that other atoms that you can use in radiometric dating as well. But the thing that's cool about this is that you can date things back in millions or billions of years based on the known rates of decay of these molecules. So this graph here shows uranium, which has a half-life of 4.5 billion years. Now that means that it takes 4.5 billion years for uranium, half of the uranium molecules in a substance, to decay into another form. So you can sort of do calculations and back calculations and figure out the relative proportion of the different types of uranium in a sample to see how long it has been since that organism died. Now this works because if you're thinking about something like carbon, you are taking carbon into your diet all the time. Plants are made up of carbon, you eat carbon in your diet, animals are made up of carbon. And so at the time that you're alive, you are representative of all the carbon that has been incorporated into your body. And so if you leave a trace of carbon, when you die, the carbon begins to decay at a regular rate. And so you can look at the amount of one version of carbon compared to another version of carbon. Now, carbon has a smaller date range, but there are different isotopes that you can use to get different distances along a time scale. So uranium was actually used to date the world or the Earth at 4.6 billion years old, based on that half-life of uranium of 4.5 billion years. So radiometric dating is used to date fossils along with relative dating based on positions in the soil. But even when we have a fossil, fossilization itself is an unlikely event, and it does not give us a complete picture of the evolutionary history of an organism. And that's because fossilization only represents a small fraction of the organisms that live on Earth. So when fossilization actually occurs, it's basically a small piece of a snapshot of a very particular time in evolutionary history. So you're only seeing a small percentage of any of the organisms that actually lived. And then on top of that, only certain structures or only certain biological remains are actually going to be preserved. And then on top of that, you only have physical structures. So you have no record of any of the biological processes that occur in a living organism. So if you're using the fossil record, you can only use the physical features that were actually fossilized at the time. Now, as an example of a well-preserved fossil record, we have the history of horses in North America. So based on the fossil record, we know that horse-like animals first appeared about 55 million years ago. And then you can follow those horses and look at the different structures, the fossils that were left from these organisms, and you can sort of trace an evolutionary history or evolutionary tree to get to the modern horse today. So these are all horses, individuals in the horse family tree that have left fossil evidence, and based on their structures you can reconstruct this sort of hypothesis of how these are all related and how we get from this ancestral form back here all the way to our modern horse today. So you may have heard of individuals looking for the missing link. Now this often refers to, when people talk about the missing link, it refers to generally human history. So looking for a link between the basically the great apes and humans, what sort of common ancestor would we have had. But there are missing links or transitional forms all over the fossil record that we haven't found yet. One that we have found is this organism called the tikalik. So this tikalik is believed to be a transitional form found in the fossil record that is a transition between fish, aquatic fish, and an animal that could walk on land. Now, if you look at this, it does sort of have some features of a fish, but it also has some features that we would recognize as something similar to organisms that spend their time both in the water and out of the water. Now, to me, this sort of looks like an alligator or a crocodile, just a little bit. But if you think about that organism's environment, they live in and out of the water. But if you look at the bones in this one group of fish called the lobe-finned fishes, which are believed to have been a common ancestor in this lineage that was animals that could walk on dry land, you see this progression of bone shape as the limbs of this organism become better adapted to walking on land. So sometimes you hit the fossil jackpot and you actually do find important transitional features. The problem is that there are often gaps. So you might have a transitional organism and not even know it, or you have no idea what the transitional organism is. But even though the fossil record is not super complete, it does still support the idea that organisms can change over time and that organisms with similar characteristics are very likely related to one another. All right, so we can also use evidence of biogeography. So biogeography is the study of how organisms are distributed around the world. So this suggests that or you can trace the origin of an organism by following sort of the patterns of where similar individuals are found. So this example here, we've got Hawaiian honey creepers. So it's believed that there was a finch-like ancestor from North America that made its way to the Hawaiian Islands. And by looking at similar features of honey creepers on the different islands, you can see how they are related, and you can also see where their common ancestor came from. Now, this is a great example of unity and diversity and adaptation, where you can see similarities to a common ancestor, but those individuals have adapted to their unique environmental pressures. So if you look at these individuals, they are all uniquely adapted, but they're also very similar to an ancestor on the mainland. And so you can trace the evolutionary history of a species based on where they are located and whether or not they're located to other similar individuals. We can also use the idea of biogeography to explain certain adaptations where we have similar features but different origins. And a great example of this are marsupials. So marsupials are animals that birth their babies into pouches, and then the babies develop there. Now, if you look at this comparison here, I find this really interesting. These are marsupials and their placental counterparts. So placental means that they have, basically they have their babies in eggs. So mammals don't lay their eggs like birds, they keep them inside, and then they birth live young from those eggs that are inside the mother. But you can use biogeography to see how these guys are related. So if all of these pouch marsupials are found in one location, they are more likely related to each other than they are related to something far away that has a similar adaptation. So we've got the sugar glider and the flying squirrel. They both have similar adaptations due to the environment that they live in, but they are not very closely related. Same thing with this numbat and a giant anteater. They have this similar face structure because they eat similar organisms. But the numbat is more closely related to the sugar glider because it is a marsupial. And we know that because they are more closely geographically related. And then, of course, we've got the Tasmanian wolf and the gray wolf in North America. And they also have similar features because they have developed in different locations, but with similar environmental pressures. So I'm going to come back to this topic in just a minute. These structures that are similar based on similar environmental features and not on common ancestor are called analogous structures. So we can also look at the anatomy of organisms and the embryonic development of organisms to support evolutionary history. So it is thought that shared embryological stages, that means shared development of the young organism, indicates a common ancestry. And the reason for this is that you can see similar structures in organisms that are not super closely related. So one of these structures is these gill pouches. So this is shown to be found in sharks, turtles, humans, and chickens. And based on the evolutionary history of animals overall, that they came from the lobed fin fishes, one lineage of which is a shark, that within the ancestral history, they all have, or the ancestor had gills, and so these gill pouches or these remnants of gills are shown within the embryo development of each of these organisms. The other is a bony tail. Now, you know, sharks have tails, turtles have little bitty tails, chickens have tail feathers with a little bitty tail that they're attached to, and humans don't have tails. But we still have the vestiges of this in our embryological development, which would suggest that we came from a common ancestor at some point that had that tail feature. Now that doesn't mean that humans evolved from chickens or chickens evolved from turtles. It just means that there's a common ancestor at some point in the history that would have had this tail structure. Now structures that we no longer use are called vestigial structures, and I'll come back to that in just a minute. So you can also look at evolutionary relationships by looking at homologous structures. So homologous structures are structures that are similar because of a common ancestry. Now they're similar because of the common ancestor, but they're different because they have adapted to different environmental pressures. So here we have the arm, and they don't show the overlay of the bones on the arm, but they're actually reasonably similar to the horse here, with the exception of the hands, which are more similar to this porpoise over here. So you've got some long bones, and then you've got a lot of bones in the hand to make them movable because we use our hands for grasping. The hand structure is going to be similar in other mammals that use their hands for grasping. The horse, the forelimb supports their weight as they're walking, so they're adapted a bit differently for that purpose. Bats have adapted their arm structure for flight. So they have these really long finger-like structures that are not used for grabbing things, but they're used as part of their wing support structure. And then we've got this porpoise who's not using its hands to grab anything, but rather the hands are part of the flipper structure that's being used to swim. And so you can trace these similar structures back to a common ancestor, which is the reason that they're similar, but they have been, even though they have a common ancestor, they have adapted to their unique uses and the unique ways that the organism needs them. So vestigial structures sometimes cause problems. Vestigial structures cause problems in understanding evolutionary history. Vestigial structures are structures that are present in organisms but no longer have a purpose. So the example here is this vampire bat has these nasty, or I'm sorry, not the nasty teeth, the molars in the front. But they have an entirely liquid diet. So they use these sharp incisors on the front to bite the animal that they are preying upon, and then they use their tongue to lick the blood. I know that's kind of gross. So these molars basically aren't doing anything because their entire diet is liquid blood. Now, the reason they still have them is that their ancestors did. So these bats would have begun utilizing this food source, but there's not really any reason to lose the molars. If they're not causing any negative impact, those molars are going to stay there. So they're there because their ancestors had them, even though they don't have them anymore. Other examples of this are hip bones in snakes. So the snake ancestors had hip bones. Even though they don't have legs attached anymore, they still have the bones. Also that tail structure, the tailbone not the tail structure, the tailbone in humans, we still have a tailbone, but we don't have a tail attached to it. So that's another vestigial structure. So sometimes you see vestigial structures in the living organisms, sometimes you see them in development. So if you go back to that embryo slide, you'll see the gill slits in humans as another vestigial structure. We don't have gills. That structure ends up developing into something very different in humans. All right, so convergent evolution is the type of evolution that leads to analogous structures. So a minute ago, I talked to you about sugar gliders and flying squirrels. Now, the reason they have that flap of skin between their forelimbs and their hind limbs is that it helps them glide in their environment. So they have both developed the similar adaptation due to similar environment. So they both live in the tops of trees. They get around by jumping between trees. And so that flap of skin helps them glide. Now, that flap of skin is not there because of a common ancestor. We saw that they were marsupials versus placental mammals. So no recent common ancestor. You'll just have to take my word for it on that one. But no recent common ancestor, but they have these structures that provide the same function. Now, this can sort of throw a wrench in deducing the evolutionary relationships of organisms, which is why you have to use some of the other things, the comparative embryology, the biogeography, in order to deduce how or even if these organisms are closely related. So analogous structures are similar structures with a similar purpose, but without a common ancestor. So another example of this is dragonfly wings, or and you could even think of bird wings, you could think of bat wings. All of these structures are similar in their shapes, not identical, but they're all similar in their shapes because they are allowing this organism to interact in a particular way with their environment. So all of these organisms fly, but their wings didn't all come from a common ancestor. They developed along these different lineages. So insects developed flight separately from reptiles, and then birds that developed flight, and then the bat developed flight separately. So bats are, I'm sure you know, more closely related to other mammals because they are mammals than they are to birds, but they have these similar structures because they perform the same function. This is called convergent evolution. All right, I know this is quick. I'm trying to get done. So we can also look at something called the molecular clock. So the molecular clock is where we get into the molecular biology of genetics. So molecular biology shows that organisms who are more closely related have common genetic sequences, and those that are more similarly related are going to have more sequences in common. So all living organisms share the same genetic code. Bacteria, archaea, eukarya all of them use DNA and a very similar process of gene expression. That suggests a single common ancestor that used DNA as its genetic code. Over time, that genetic code has changed as organisms have populations have adapted to their environment. But those individuals who are more closely related are going to share more similarity in their DNA sequence to each other than they are to those individuals who are less closely related. So you and I are going to share more DNA than, say, us and a chimpanzee. But humans and chimpanzees are also going to share more DNA similarities than us and a bird, something like that. Or and we're going to share more similarities between us and our pet dog than we are with a particular bacteria, because we're more closely related. So this similarity can be measured by looking at the DNA sequence and actually counting the differences. And from that, we can use that information to be able to deduce some evolutionary relationships between two species. Now, this is generally done by comparing specific genes. So this example here is the hemoglobin gene. So we've talked about hemoglobin quite a bit. This is the one that is in our red blood cells that carries oxygen, the one that in its mutated form produces sickle cell. So if we use humans as our reference sequence, because that's us, we like to talk about ourselves, we have 146 amino acids in our hemoglobin, and so there's a specific sequence that codes for that amino acid sequence. If we look at chimpanzees, we have 8 amino acids different. So hemoglobin in humans and humans and rhesus monkeys are much more similar than the other organisms. As we move away, we look at another mammal, but one that's not very closely related, 32 amino acids. Going out to the bird, whose common ancestor was 112 million years ago, 45 differences. So over time, these species have accumulated differences since the common ancestor. Over here, we've got the lamprey. The lamprey is sort of thought as one of the very earliest animals, and so it is, if you look at an evolutionary tree, it's often the first branch off of the animal side of the tree, and you can see that the majority of hemoglobin amino acids are different than human hemoglobin, suggesting that through this evolutionary history, the hemoglobin has changed, and now it is vastly different between humans and the lamprey that had a common ancestor estimated at 564 million years ago. And this is because each of these lineages has been evolving separately from the split from that from that common ancestor. And so they've been accumulating these genetic differences along the way. So finally, we get to experimental evidence. So we've got these other lines of evidence, different ways that we can support evolution. I would say the strongest one is being able to see evolution in action. See how a population of organisms can change over time. So we've got lots of laboratory and field experiments that are going to support this idea of evolution. So this is kind of a cool study here. This was a study that was done on grass at a golf course. So really, you can study anything you want. So based on the different ways that grass is cut at a golf course, they looked at selective pressure and the change in this population over time. So the grass on the putting green right around the flag there is always kept really short. And this is so it doesn't interfere with the roll of the ball. On the fairway, it's a little bit longer, so it's allowed to grow a little bit longer before they cut it. So you can see that mowing happens much less frequently on the fairway grass than the putting green grass. And then outside in the rough, they let it go much, much longer. According to this graph, they cut it once a month. So they looked at how fast this grass matured and produced seeds and how many seeds they produced. And they found that over a few years, grass that was continuously cut produced many more seeds much more quickly than these other ones, because it had to get out the seeds before it was going to be cut, because otherwise they wouldn't be able to pass on their offspring. So the grass in this area that was already able to produce seed more quickly was selected for. So if you start with the same species of grass throughout this entire putting green, you are of the entire course, not the putting green, you can see that there's been a change, a genetic change in this population that has allowed some of them to sexually mature and produce higher seed rates much more quickly. So I think the best evidence for supporting evolution is the idea of antibiotic resistance. So in antibiotic resistance, you get the development of bacteria being able to withstand basically medicines that are used in hospitals. So in the 1940s, penicillin was discovered, and it was used as the first antibiotic, and it was effectively able to kill Staphylococcus aureus. So this is the one that can cause blood poisoning. It causes something called MRSA, if you're familiar with that, in hospitals. And the reason for this is in the 1940s, it was extensively used. Penicillin was extensively used, and it was used so much that at some point, some bacteria developed a mutation that allowed them to survive in the presence of penicillin. And so this is what we see here. We have some Staph aureus that is able to survive even though there's penicillin. So these guys on the outside edge probably are being exposed to less penicillin, and so because the penicillin is on this disc and spreading outward. So those that could survive a small amount of penicillin were actually able to pass on that genetic information to the next generation. And so over time, the effectiveness of penicillin went down. Now, penicillin isn't the only one that antibiotic resistance has come about for. Many antibiotics show resistance. MRSA is a common problem in hospitals. It's called methicillin-resistant Staphylococcus aureus, so it's the same bacteria. And it is, multiple resistance. Sorry, not methicillin, multiple resistant. It means that these Staph aureus have developed resistance to multiple antibiotics. Now, this is really important because even though bacteria reproduce asexually by binary fission, they can actually transfer genetic information to other bacteria. So they're not only transmitting vertically from parent cell to daughter cells, but they can also transfer bacterial resistance horizontally. So they can pass the antibiotic resistance to closely related species, sometimes not so closely related species, and other bacteria within their same population. So there's a lot of evidence for supporting evolution. We saw that fruit fly experiment in the very beginning. And there's a lot of examples of natural selection. And if you watch a population, just observe them over a long period of time, you will see that population change in response to a particular environmental pressure.